All right, everybody, we're going to discuss photosynthesis today. We've already done a little bit about the physics of light and some energy stuff, which is in one of my other lectures, okay? So we are looking at this. This is my first time doing a camera video. I usually use my webcam, so I'm trying something different today. Hopefully you can see up here. If not, this is all my website, so follow along with what I'm saying. Keep this up in a different window, and I think you'll be good. You people in class, your guided notes I gave you, which if you're online, the guided notes are posted in this week's folder. So you people in class, the guided notes don't start with the first blank. They're going to start a little bit in because some of those guided notes go with the lecture 1. This is lecture 2 of photosynthesis. There'll be 3 of them. So just starting off, photosynthesis in AP biology is one of the harder topics. People say unit 3, which is where we are right now, and unit 6 are the hardest. Unit 3, pretty easy with the enzymes. You guys have been following along with that really well. The energy ideas, the idea of thermodynamics, most people do really well with that. But what we're probably going to struggle with is learning this in one shot. A lot of students are very, very talented and they can see something like this one time in class typically, but most of the time people need to look at this 2 or 3 times. So this is recorded. You can watch me do it a couple times. I recommend you watch my stuff, but I'd like you to look at what other people put out there too. So you're probably going to have to watch a couple different videos. You're probably going to have to do a couple activities to keep track of what's going on with this, because there's a lot of moving parts and pieces that happen at the same time. And today we're only going to cover the light reactions, and then we're going to cover the Calvin cycle next. So let's get into it. Now, all this stuff is happening in the chloroplast. Really big overview. So far in biology, you've probably looked at the chloroplast as a black box. And you probably know that light and water go in, and CO2, and sugar comes out, okay? You probably know that. But it's way more complex than that. We're gonna kind of get into this diagram down here, which looks really terrible. You don't have to memorize this whole thing. You just have to know a couple parts and pieces of it. So we're going to talk about that. Make sure you're in your guided notes, probably following along right now. If you were a botanist and studying this on a much, much more detailed level, it would even go deeper than this. So we're going to kind of take that black box, open it, look inside of it, and see what sort of the first level of that is. So everything here happens in the thylakoid, which remember are these things that kind of look like green cookies stacked up. So all this is taking place in the thylakoid. Now look, I want you to, your eyes up here for a second. These are phospholipid bilayers right here. Those phospholipid bilayers are not the cell membrane. They're not the chloroplast membrane. They are the thylakoid membrane, which some people call the thylakoid lumen down here. So don't get lost because there's phospholipid bilayers in lots of different places. This is why we did enzymes first, because there's a lot of enzymes working here. Enzymes work best when they're compartmentalized in areas that do specialized functions. So what you're seeing here is an example of that. That's why we talked about that first. Now we're going to look at it in photosynthesis, and then when we're done with photosynthesis, we'll do respiration. Okay, so everybody knows this stuff up here. That, you know, grade school kids know that solar energy is converted to, they'll probably say food, but we're going to call it chemical energy in the form of, right now, for this side of things, ATP, and a new molecule called NADPH, which we'll talk about in a minute. Okay, so we're going to start with what are called light reactions because they use light. Okay, some people used to call the other one the dark reaction, but now we call it the Calvin cycle. In light reaction, energy is made into two different currencies, two different kinds of things. Okay, I see you out there. You finding it? Okay, good. A couple people in class having trouble finding where we are with the guided notes. So chemical energy comes in these two forms I talked about on the last slide, NADPH, and that stands for something that I can't remember. It starts with nicotine. You can look that up if you want to. It's very, very long, and that's why we abbreviate these things. There's a lot of abbreviations in this area. You're going to look at some things called plastiquinone. You don't have to know those. Plasticinin. Like, you don't have to know all these. The ones you have to know, I'll tell you. There's a lot going on here that we're not going to get all the way down to that level, okay? So how does this happen? So what happens is the cell gets this done by using photons to excite electrons. And some of you are chemistry people. Does anybody remember energy levels and electrons jumping energy levels and coming back down?
Oh, yeah.
Okay, I see some people saying yes. I see some heads nodding. So when there are certain conditions where an electron will jump up an energy level, then when it falls back down, it goes into some excited state when it jumps that energy level and it comes back down. A lot of times it will release light. So let's take a look at how that works. So you've got this chlorophyll molecule, and then you have the electron jumping up and down. Let's look at what's going on there. First, the chlorophyll absorbs a photon of light. What percent of sunlight under perfect conditions do you think a plant can convert? Just give me a shout out.
1%.
1% is pretty close. It's about 1 to 3%, which if you take like solar panels on a house, they're way, way better than that, okay? But that 1 to 3% is responsible for all life on Earth. So it doesn't have to get all of it. It just gets about 1 to 3%. So what happens here? You guys remember what an E negative is, right? We can use that shorthand without anybody getting confused. That's an electron. The electron is boosted from a ground state. That's sort of closer to the nucleus. It goes into this excited state where it's not very stable, and it falls back down. When it falls back down, it releases energy as heat, and it releases photons, okay, as fluorescence or glow. Okay, you've probably heard of fluorescent lights. So we're going to do something with that. That energy, that falling back down, is what we're interested in. That gives us sort of like a potential energy. All right, let's move on. Too fast? Are you okay? Everybody keeping up? All right, so there's a couple things going down here. Look, we're simplifying this now. There is what we call different photosystems. And photosystems are reaction centers. They're membrane proteins. Look here, membrane proteins. Where's this membrane? Remind me. This is the membrane of the thylakoid, okay? In the thylakoid membrane protein, there are two photosystems that do this, two membrane proteins that do this. We're going to talk about them. And so what's going on inside of this PS1 and PS1 and PS2 are chlorophylls and electron acceptors. Okay, so I'm going to zoom in on these in a little bit. They're going to be boxes because those are easy to draw with the basic graphics tools in these lectures. So let's take a look. There are light capturing complexes. Those are pigments that are part of these proteins. Now, if I took apart an iPad or a cell phone, how many are there? Is there one antenna in there or multiple antennas? There's multiple. What are some antennas that are probably in there? How do you connect to your AirPods and things like that? Bluetooth? There's going to be a Bluetooth antenna. What about if you're going to be a Wi-Fi antenna? What if you're out on the cellular antenna? Why are they different? Yes, they're different frequencies. Antennas are designed with different frequencies. So think of these like as antennas to not send, to absorb radio waves. Back before cable, you had to put a TV antenna up on the roof, okay? And there were different antennas for different parts of the TV stations, okay? So these are tuned or specifically set up to absorb certain frequencies of light. Now those frequencies are very close to one another, okay? Let's break these down. There's two photosystems, PS1, PS2. Okay, we're going to start with photosystem 2. Now, this is one of these things in science that all students hate. Photosystem 2 is where things start, and photosystem 1 is where they end. And you're probably thinking, well, why'd they name them that way? Why didn't they say the first thing that in this cycle should be number 1? Well, here's what happened. They discovered photosystem 1 first. And then later on, they discovered, well, we'll call this one photosystem 2. I imagine they didn't call it 1 when they discovered it. So we're going to start the reaction over here and move toward photosystem 1. This is in order for students to learn it. In a real cell, these things would be arranged all over the place. So we're going to go from left to right. That means you start with photosystem 2. It just so happens photosystem 2 absorbs the wavelength of about 680 nanometers, and photosystem 1 is about 700 nanometers. So two different wavelengths or frequencies. All right. So named in order of discovery. I already talked about that, but we're flashing it up on the screen for you. All right, let's move on. So, the next couple slides, I want you to pay attention to where the electrons are. This is intimidating. If you look up here, there's lots of steps. You don't have to know every single one of them. You basically need to know photosystem 2, cytochrome, photosystem 1, and then ATP synthase. These three right here are the really important ones. Now there's a thing that goes between these, and there are things happening up here. You also have to know this. Now, the people that designed these AP test questions, how many questions do you think they could derive from this? A lot. And they can get really sort of like, what would happen if there was a malfunction in photosystem 1? You would have to like be very specific. They're going to ask you that to see if you can tell. They're going to ask you that so they can tell if you really understand this or not. Those are the typical, like, what would happen if AP questions, and there's a lot of ifs that could happen up there. All right, now look, this is photosystem 2. Let me back up a little bit. I'm taking this membrane protein, and I'm representing it with this box. Okay, what does this box have in it? It's got some pigments, and it's got an electron acceptor. Okay, pay attention to what happens here. You can see why this is hard. You can see why you're going to have to look at this more than one time. First things first, it's a photosystem. That means light. Okay? Light hits the pigments. So what's happening here is that photon causes an electron to go into an excited state, and then it drops back down to ground state, and it keeps doing that until a pair of these 680 chlorophyll A molecules carry that energy. Watch. So the light carries that until that electron gets excited to the point where it gets accepted by what's called the primary acceptor. So once it gets enough energy, the electron is carried up to what's called the primary acceptor. Now, you do not have to know that that thing's called P680 plus. All you have to know is that we have excited electrons. Okay, excited electrons. Now, something else is going to enter the system here. Water. Okay? Water is going to now have its electrons pulled off, and it's going to split into hydrogen, and then it says half of an O2. That can be a little confusing. Why is it half of an O2? Well, there's only one oxygen here. One oxygen is half of 2. So each water donates half of an O2 and 2 hydrogens. So what do you think is going to happen to the next water? What's the oxygen on the next water going to do? It's going to immediately join with one of the other free oxygen molecules, and it's going to make O2 really, really quick. So don't think of this as like, well, there's just one water. They just want you to see, well, what happens to each water. Each single water molecule gives half of the reactants to make an O2. So this is a common point they might ask you. Photosynthesis splits water. Is that photosystem 1 or 2? So you would say what? Photosystem 2. The oxygen released by plants into the atmosphere, where did it come from? What input did plants make atmospheric oxygen from? You would say water, okay? Because plants are taking in more than just water. They're also taking in CO2. So what are the big inputs for photosystem 2? Light and water. Light and water, photosystem 2, light and water. All right, like I said, this is confusing. You might have to look at different diagrams. You might have to look at other videos. You're going to probably have to do some practice questions. Stuff like this, very, very hard to learn just seeing it one time and have one person explain it to you. Look at different people. So, I already told you this, but just to review it, O2 is split into its negative electrons, and then you've got these 2 hydrogen ions. What exactly is a hydrogen ion? A positive hydrogen ion. Like, what's in the nucleus? It's just one proton. So if I say proton or an H+, those are the same things. Okay. So then we get these two. So we get the 2 hydrogens out. Where the hydrogens go is important. Are they inside the thylakoid or outside the thylakoid? They're going to be inside of it. Okay. And we already talked about the oxygen, and it immediately bonds to a second oxygen. Okay. So after these electrons are stripped off, where they're going to go into what's called the electron transport chain. That's a lot to say, so we call it the ETC, the electron transport chain. And that electron transport chain, we'll see a little bit later, but it's actually, here's my membrane. The electrons actually travel in between the phospholipid bilayer of the thylakoid membrane. So where do these electrons go? They go to photosystem 1 in the electron transport chain. Where's that at? It's in between the phospholipids. So I want you to kind of know where everything's happening, because that's something you're going to be expected to maybe explain later. Now this PQ, these are intermediate electron carriers you don't have to know. Cytochrome, I like for you to know where that's at. Just know that cytochrome is in the middle of the electron transport chain. They call it a chain because chains have a lot of what? Links, and each link is actually really, really complex. We're not going to get down to like what the names of these are. Just know that cytochrome's in the middle. We'll talk about that. Okay, there's another link in the chain. All right, so I know people on camera probably are having trouble reading these words. You can kind of see right here, there's a loop. The electrons are the blue part, and they're coming over to cytochrome. They're coming over to photosystem 1. But let's pay attention here. Really pay attention to this, everybody. Water came in and water got split. This is the inside, called the lumen, or the hollow space inside the thylakoid, which is like the green stack of cookies. The stroma is sort of the cytoplasm of the chloroplast, okay? So photosystem 2 concentrates hydrogen where? Inside or outside the thylakoid? Go ahead, guys. This is the lumen, so it concentrates hydrogen. Somebody say it. Come on. Inside the thylakoid. Inside the thylakoid. So look, light and water. Water's being split. Hydrogen's building up. Hydrogen ions are building up inside of the thylakoid lumen. Why don't these hydrogens immediately form H2? They don't have electrons. The electrons have been stripped off of them. So let me ask you this. Knowing what you know about cellular transport, that was back in Unit 2. If this wall is a membrane and something's piling up on the other side of it, in cells, does it want to come through? Yes, okay? If I can make what's on the other side of this wall build up, that's a form of kinetic or potential energy? Potential. If that hydrogen can go through a special protein all the way over here, when the hydrogen gets pushed through that protein, this thing actually spins around like a windmill, and that's what makes ATP. So I'm holding back the hydrogen. It comes through the membrane because it has potential. It's trying to reach equilibrium. When it comes through the membrane, it spins this enzyme over here and turns that ADP, which is spent ATP, back into ATP. So photosystem 2, cytochrome, photosystem 1, ATP synthase, powered by that hydrogen potential, basically all the hydrogens building up inside of thylakoids. That is what makes ATP for the Calvin cycle to make glucose. Do you see how this is getting really complex? And there's a lot to absorb here. You can really see it, okay? So this is, I don't like to make long lectures, but there are some things you just can't simplify. So just to review, we're stripping electrons off of water. We're putting hydrogen inside of the thylakoid. Thylakoid is holding it back. When it passes through the thylakoid membrane, it turns ADP into ATP. Now, other things are going to happen simultaneously in photosystem 1. So let's review what I just told you. Basically, the fall of electrons, and when I say fall, I mean going from that high energy state to that low energy state. From photosystem 1 to 2 gives you the energy to make ATP. So this is a big deal. Turning light from the sun into energy, really big deal. No life would be possible on Earth without it. It's certain frequencies that have come through our atmosphere. There's a lot to this, a lot of steps. Okay, so what we get is what I was talking about when I say hold back the wall. We get a hydrogen gradient. Hydrogen piled up on one side of the thylakoid gives that potential. It's almost like a dam holding back a river. And when hydrogen goes through a water wheel, it can spin that enzyme up. It literally spins around like a merry-go-round, and it turns ADP into ATP. I know I'm repeating it, but that's because it's complex. And the name of that enzyme, it ends in ACE, is ATP synthase. Don't confuse it with ATPase. That's what breaks down ATP. So synthesize means to make. All right. We're going to jump to photosystem 2. Okay? Photosystem 2 gets the electrons that have passed through the membrane. Okay? They go through cytochrome and they are accepted. What are we doing with the electrons? We're getting them up to that energized state, and we're doing that with light again. So plants can use 2 different frequencies of light, which can be a handy adaptation to have, okay? You can see same arrows we had before. Right here's where we want to pay attention to. We get electrons into an energized state. When they're up in that energized state, they go to what's called the primary acceptor. All right. Well, what is that energy going to do? When that energy comes back down, they go to a second electron transport chain. And the one thing I don't like is I wish this was on the top so that you knew this was happening outside the thylakoid. So what we have now is another substance. It's ending in ACE. What's that mean about this thing? It's an enzyme, okay? So what does it do? NADP is a hydrogen carrier, and it gets reused. So NADP joins with these hydrogen ions. Now, this is kind of cool, and this is why biologists geek out about this, because it's so elegant. It reuses the hydrogen. Where did this hydrogen come from? Go all the way back to the water. The water gets split in half. The hydrogen goes through the enzyme. Now that it's on the outside of the cell, the hydrogen's going to join with NADP, and this enzyme is going to turn it into NADPH. This molecule is a hydrogen carrier. This stands for phosphate. This is hydrogen. You don't have to know what it stands for. But we're going to carry this hydrogen over to the Calvin cycle, and this actually has chemical potential energy in it that's going to be used to make what? What's the ultimate product of photosynthesis? Sugar. We're not going to get to the Calvin cycle in this lecture. Okay, so let's put it all together here, all right? You've got light coming in. You've got water here, okay? What ingredient are we not dealing with at all in the photosystems? Like photosynthesis takes 2 ingredients, well 3, light, water, and CO2. No CO2 on this side. CO2 comes from the carbon dioxide over in the Calvin cycle. So you have to be able to separate these out. Like, what are the ingredients of this one? What are the products of this one? What are the ingredients of the Calvin cycle? What are the products of the Calvin cycle? So we're not going to talk about that one today. The only thing we're doing in photosystem 1 and 2, or the light reactions, is splitting water, okay? So we have water come in. In photosystem 2, it gets split in half. It's going to make oxygen. It's going to make hydrogen ions. The hydrogen ions are building up inside the thylakoid lumen. They pass through ATP synthase, and they make ADP, which is like spent ATP. I always kind of use this toy up here to say this is ADP, this is ATP, putting back together. Well, how does it get put back together? Well, in plants, this is what we're looking at. This is how it happens. Okay? At the same time, we have things going through. What's this thing right here where electrons are moving through? What's it called? You guys are shy on me. Electron transport chain. So there are many links in the chain. You don't have to know them all. You just have to know that electrons move in between the phospholipids, then get moved to photosystem 1, where they're excited again. The same electrons that got ripped off over here are going to be excited over here. Now, what are these guys going to do? The light energy that excites these electrons, you don't have to know these right here, basically turns NADP into NADPH. So that is a summary of the light reactions of photosynthesis. These are the things you really need to know, okay? We have over here, you've got water coming in, ADP and NADPH. These two get recharged. These are basically rearranged versions of the inputs. So is this a low energy or a high energy? This is low energy. We're going to use an anabolic process to put this back together. Okay, is that exergonic or endergonic? Endergonic. We're going to do the same thing here. Okay, the water, we're really just using the hydrogens. After the hydrogens are stripped away, oxygen is a waste product. So know these. Know where each one of these happens. Each thing's happening in a different spot. Each one of these products is going to a different place. So if I said, is the ATP inside or outside of the thylakoid? That's going to be outside. Is the hydrogen that gets split apart, where does it build up? Inside or outside of the thylakoid? Inside. How does it get out? It goes through ATP synthase. Okay? Where's this NADP at? That's outside the thylakoid. The hydrogens that were once part of the water get hooked onto NADP and make NADPH. All right. Summarize this a little bit more in written form, okay? So, starting off, this is what most, even people in elementary school know here. Light converts, plants convert light into chemical energy. Most people can say that, but most people cannot say, well, what does that even mean? Well, it makes two molecules that are energy carriers through anabolism, okay? NADPH and ATP. Well, how does it do that? Well, it splits water. Water provides electrons and protons. We don't really care about the oxygen. But once we split the water in half, we split the hydrogen in half too. Well, how do we do that? Well, the light that's absorbed by chlorophyll pushes these around to form an electron acceptor and a method to recharge ATP. Okay, it phosphorylates ADP. That means you add a phosphate to it. Okay. That's a lot to get to. So this is the week before Thanksgiving break. We probably won't get the Calvin cycle done. During break, I do want you to take a break, okay? So I will probably spend the rest of the time we have together before break really hitting you hard on all this stuff that's happening in the light reactions.